So good morning still, everybody. It's my pleasure to present Cristal Therapeutics to you. And we're developing what we call cryptic nanomedicines for superior therapeutic profiles. And actually the key need that we're trying to address is that chemotherapeutic agents, either existing ones or new ones, they have either issues on the safety profile or on the efficacy profile, namely safety to too much adverse event, either as a monotherapy or restricting the combination regimens, or they have insufficient exposure to the molecular targets. And with our cryptic nanomedicines, we have already clinically validated that we have either an improved safety profile. So in this case, we have the dose-limited toxicity of neutropenia significantly dropping as compared to the comparator drug, or we have much higher uptake in the tumor tissue, clinically validated. But I'll elaborate on the details later on clearly. Because Cristal Therapeutics, we're a small company. We're located in the Netherlands. We're 15 people only, but we are already phase 2 stage. And what we're developing is what we call, as said, the cryptic platform. So nanoparticles that can entrap a broad range of therapeutic agents, from small molecules to peptides, and we're currently also expanding it to oligos. But the key thing is it's clinically validated, and we aim to develop not only a product ourselves, but also in co-development with other companies. So we're not a service provider, but we really aim for co-development in here. And we means we have a strong management team, as well as that we're financed by private equity. So recent years we raised up to $30 million over time. And the management team is comprising not only of myself being the founder and the CSO, but together with Axel Mescheder, who's our CMO and CEO, who has a really wealth of experience in both the medical field, as well as also Henry Verwey, who's the CFO, and he also have been very successful in raising money, as well as going to IPOs in both Euronext as well as Nasdaq. So then let's talk about the underlying science. What we take advantage of is the fact that in tumor tissue, when you have rapid growth, as it calls angiogenesis, there are, as we call it, leaky vasculature. So meaning the endothelial wall is not as nicely aligned as what's happening in the healthy tissue, but rather there are small gaps, and actually we take advantage of these gaps. So we have nanoparticles that are long circulating in the bloodstream. Here there is no passive diffusion, there is only active uptake of compounds of the bloodstream, for example glucose, that those compounds are actively taking up. But in the tumor tissue, we can take advantage of these small pores by having a nanoparticle long circulating, and then it can extravasate into the tumor tissue. But then clearly in the tumor tissue, it has to release its cargo, otherwise it wouldn't be efficacious. So the way how we embed that is by making these type of what we call polymeric nanoparticles. So I do not know to what extent you're familiar with liposomes. Liposomes are physical structures, but in our case it's different. Namely that we initially have a drug, we covalently link it with a linker molecule. If we then combine it with a polymer, then we create these type of 3D structures, which is actually one big network, because in our case, both the drug molecules as well as the polymers, they're all covalently linked. So that really assures a good stability if you administer into the bloodstream, and then thereby also really having good tumor uptake. So this is all patent protected by patents owned by the company. And actually what we've done, as said, we developed a product all the way to the clinic, meaning more specifically we entrap Docetaxel in there. Docetaxel is part of the taxane class. I do not know to what extent you're familiar with that, but taxanes remain to be one of the cornerstones of chemotherapy these days, even despite the developments in immuno-oncology. But what is the fact is that the taxane in general, particularly in recent years, Abraxane, which is a nanoparticulate construct, is having really significant sales. So it's really the blockbuster compound, and they expect it to be further growing in sales in recent years. But the key thing is there is not yet a Docetaxel product on the market, and that's where we thought there is an opportunity for us. So Taxotere is the Docetaxel innovator product, as we said, and while we profile ourselves as compared to Taxotere, in this product profile, with the key things being either reduced toxicity, so really having a better profile, but also having more exposure to the tumor tissue over time. And clearly there are also some regulatory requirements as well as some manufacturing essential requirements. But I'll immediately like to guide you to the clinical data that we generated, and what we've done actually is we're taking these nanoparticles, and you can either take tumor tissue out it, but what would be more elegant to literally label the particles with a PET label, enabling for non-invasive imaging. And that's what we actually done. So we label them, that allows for imaging via PET scanning, with in this case zirconium labeling, so that enables scanning up to at least 1 week. And then we monitor the biodistribution in the patient setting, and this is after 2 days or after 5 days. And you literally see, as indicated here, very long circulation in the bloodstream. And then penultimately also you see the main organs where the particles will accumulate. Well, in this case, clearly the main organs are the liver and the spleen, which is kind of known for these type of particles. But I would say the most important part is clearly, do these nanoparticles reach the tumor tissue, yes or not? Because this enhanced permeation is kind of debatable in the preclinical setting, and in the clinical setting it has hardly been evaluated. So I'm pleased to share these data with you, namely that if you take a look where the tumor located, that's what's quantified by FDG PET scanning. So here are clearly the tumor lesions. Then if you do CT scanning, you take a look, well, really these are the tumor lesions themselves. And then if we measure where is the zirconium, so where are the nanoparticles actually residing, you see a fairly good overlap with the tumor location. So indicative, also in the clinical setting, our nanoparticles do reach the tumor tissue. And then next to that, we've then done quite some additional studies to illustrate the clinical profile. So for example, taking a look at the PK profile, and I think the picture speaks for itself. So the nanoparticle circulates much longer in the bloodstream as compared to the innovator product. That results in this much more improved safety profile as indicated. And also if we take a look at the absolute quantification of the tumor uptake over time, well, I think the figure speaks for itself, more than 4-fold uptake as compared to the Docetaxel. So we've done several phase 1 studies, as well as currently we're working on a phase 2 study where we have as a primary indication ovarian cancer. We expect results to be available by summertime. So we have treated almost more than 80 patients so far, really showing the improved safety profile and now testing clearly the efficacy in the small phase 2 study. And in sense of a regulatory trial, we now aim to go forward in castrate-resistant prostate cancer. And from the regulatory perspective, we aim to develop this product via 505(b)(2), so the hybrid model, as they say. We have already good confirmation from both the European as well as the FDA agency really support our plans and our strategy in this regard. So with that, I hope to have shown you that the CRIPIC composition as such, being kind of the pharmaceutical composition, can really translate it to superior clinical profile, so superior pharmacokinetics as well as tumor uptake, but at the same time also showing an improved safety profile. And not unimportantly, but at the end of the day, we should also be capable of producing the material, and that's what we know, that the cost of goods and also the manufacturability is really straightforward. So the CPC634 is a showcase of the platform. But what we've done and worked on in recent years is that we've also kind of showed the broadly applicability of the platform in general, meaning that we have demonstrated broadly applicable for a whole variety of modalities. We have full control over the particle size, the release kinetics. We have extensive characterization in-house available. And as said, we have the material already upscaled to large-scale production. And all in all, the results consistently demonstrate that at the end of the day, a certain biological exposure is required. We can translate that back into the nanoparticle composition, so enabling really rational design. And we demonstrate for the various composition, we have good uptake into the nanoparticles of the drug. We really show in vivo the significant difference PK profile. So this is the nanoparticle, this is again the free drug. And we have really selective release depending on a certain exposure that one likes to have with this API. And we demonstrated this with a whole range of molecules. I said small molecules, either the more classical chemotherapeutic agent as well as also STING agonist, TLR, so immuno-oncology agents, as well as also with a couple of peptides, in either in oncology or even outside oncology. And with having said that, I hope I've shown you that the CPC is the in-licensing opportunity that we like to offer, which is really showing already excellent therapeutic potential with a clear path forward towards market registration. But more in general, we also like to offer the CRIPIC platform in a co-development approach where we really like to develop new products in oncology or even outside. And with that, I'd like to thank you for your attention.